Chapter 3, Basic Principles and Concepts, Immunology and Pathology, Adaptive Immune Responses of Vertebrates, Part 1. When we're discussing the immune response, specifically the adaptive immune response, unlike the innate's responding to anything not you, okay? As long as it has, something that'll trigger, a complement receptor, a scavenge receptor, one of the, how it has a PAMP and interacts with the PRR receptors, whatever. The adaptive immune response, the B cells and T cells, is a fine-tuned system. It's a fine-tuned system that responds not to the bacteria, not to the virus, not to, the parasitic worm. What it responds to is an antigen. And an antigen is a protein, a glycoprotein, a glycolipid, something that will elicit an antibody generation. What does that mean when I say the antigen is a glycoprotein, a glycolipid, or a protein? Well, on the surface of the bacterial cell will be millions and millions, if not billions, of different glycoproteins, glycolipids. On the surface of the virus, on the surface of the parasitic worm, same thing. These glycolipids, these glycoproteins, are of a set shape, a set structure that is inherently foreign compared to what you will find in your body. So that is what the B cell or the T cell learns. It learns to respond to the presence of part of a protein, part of a glycolipid, part of a glycoprotein on the surface of a bacteria, a virus, or parasitic worm, not the organism itself, part of something. What normally occurs is the body has to somehow present to the adaptive immune system some antigen that's not you. Because the B cells and the T cells during early development, the early stages of development, the early stages of maturation in the bone marrow, they were chosen against, they were punished for any cell that recognizes self. If a cell recognized itself, it was immediately destroyed. So the only B cells and T cells that would survive the bone marrow were ones that totally ignored any protein, glycoprotein, glycolipid that may be found on one of your own cells. So that way, the B cells, the T cells that get out of the blood, circulate in the bloodstream, and then find themselves housed in the lymph nodes, the jaw, the armpits, the abdominal cavity, the groin, wherever. None of those B cells, none of those T cells will ever react to your own proteins. They will not react to self. They will only react to non-self, anything foreign that is now somehow inside your body. Well, that protein, glycoprotein, glycolipid, whatever, has to somehow get from somewhere in your tissues to the B cells and the T cells that are housed in the lymph nodes. Well, first step is any cells surrounding the wound, any cells surrounding that foreign organism are going to take up the bacteria, the virus, take up parts of the parasite that may have come off, or if it's shedding as it's developing from different larval stages, will take up in a phagosome, use a lysosome to digest the stuff down, and then it's going to present it. Boom. Here you go. But the problem is that's an epithelial cell stuck right there. Or it's a macrophage that's passing by. Somehow that macrophage has to get that chunk of foreign antigen, has to get it up here to the B cell or the T cell, and then present it to it. If it's one of the, epithelial cells here or endothelial cells in your gastrointestinal tract or wherever, the way they present it, say here's something, is via major histocompatibility complex, MHC. MHC is how all the cells of your body will present proteins. Here's part of a protein. Here's part of a glycolipid. Here's part of a glycoprotein. So that way, any traveling by dendritic cell or any B cell, T cell, macrophage, or whatever, going by can look. Is it self? Is it not self? If it's self, that means those cells there are healthy. There's nothing going wrong around them. If it's not self, it's going to start a whole cascade, a whole new type of response that we call inflammation, and we'll talk in a later lecture. Now, it turns out there's two types of major histocompatibility complexes. Type 1, this MHC is going to be found on every cell of your body. Okay? Type 1, its job is just to present out. Here's something to review. Here's something to review. It's kind of like, when you're working with somebody and they're not sure what they're doing. They're constantly going to be looking at you for reassurance. Am I doing this right? Is this correct? Does this look good to you? Well, that's what these cells are doing with the MHC1. Constantly putting stuff out. Because once in a great while, it may put out something that's not okay. Something not you, which is going to trigger the immune response. On the lymphocytes and the macrophages, you're going to find what is known as class 2. Remember, macrophages are constantly looking for anything. So if they come across something that's going to trigger a scavenger receptor or one of the PRRs because it's a PAMP, that immediately means you have an infection. Macrophage is going to take in, digest, and it's not going to completely break down all the proteins and glycolipids and glycoproteins, because portions of them, it's going to put it into an MHC2 and then on its surface so that it can go to the nearest lymph node and say, Mr. B cell, have you ever seen this? Mr. B cell, have you ever seen this? Mr. T cell, Mr. T cell. Until some B cell or some T cell says, yes, I recognize that. I will now go and kill it or start making antibodies against it. So when you look at the pictures down here at the bottom, see those? They're colorful looking pictures. It's an artist rendition of what MHC1, MHC2 looks like, class 1, class 2. Notice, the different regions that are shown there. Those different regions are immunoglobulin regions. Okay, these are conserved regions between MHC, different receptors. We're going to talk in one of the next lectures. B cell receptors, T cell receptors. They're also going to be similar to what we're going to talk about in this lecture, antibodies, hence the name immunoglobulin domains. But it's at the bottom. You look down there at the bottom, you'll see that there's either one or two transmembrane portions. Hence the name, class 1, class 2. Where the two parts meet, you'll see there's a little cleft right there. In that little cleft, that's where the part of a protein, the part of a glycolipid, the part of a glycoprotein will sit so that the cell can now go, here you go, do you see this? Do you recognize this? Do you recognize this? So we know MHC. Now let's talk antibodies or immunoglobulins. When you look at the antibodies, you'll notice that the base structure you'll see for all antibodies is this quasi-Y shape. Okay, this quasi-Y shape that at the ends of the Y there will be what is called variable regions. The amino acids that make up those variable regions are going to, well, as the name says, be highly variable. Different antibodies from different B cells are going to have different amino acid sequences there because they're going to interact with different antigens. Okay? The variable sequence is going to interact with the antigen. So the ends of the Y is going to interact with the antigen. And it has to have the correct shape in the variable region to interact with the antigen. If it doesn't have the correct shape, there's no interaction. It can't bind, and that's the job of antibodies, is to bind and neutralize, bind, opsonize, bind and aid in uptake by macrophages, neutrophils, or any phagocytic cell. You'll also notice at the bottom of the Y, from the base of the Y arms down to the bottom, this is called the constant region. Okay? The constant region doesn't change. Okay? Doesn't matter which B cell lineage it came out of. It will always have pretty much the same amino acid sequence. Same amino acid sequence, which means the same shape, same structure. Because it has the same shape, same structure, you can have one type of FC receptor, or the one constant receptor, antibody constant receptor on all phagocytic cells. Variable region is going to be different, B cell lineage to B cell lineage. Okay, because the antibodies made by one B cell may not react with antigens present, but there's always a chance that somewhere in the millions, if not billions, of B cells in your body, there's going to be one somewhere that can react to that antigen. Well, to react to it means it has to have a variable region that can bind. We also describe antibodies not just by their variable region and their constant region, but by heavy chain, light chain. You look over here, also by their Fab fragment, and as I alluded to just a second ago on the last slide, the FC portion. The Fab is what happens when you digest down. I use a special enzyme to go and cut where the Y arms meet. Cut those off. They call that a hinge region. Cut there, and you end up with antibody fragments that can bind to antigens. The FC portion contains the majority of the constant region. This FC portion is going to be pretty much the same no matter which B cell's producing it. Hence the, as I just alluded to on the last slide, FC receptor. 5 classes of antibodies. IG, immunoglobulin, then it's MGAED. And if you look right there in the middle, you'll see the different shapes they have. M has that penta antibody shape. Yes, that's exactly what it looks like, that snowflake-looking thing. If you were to take immunology, you would learn that the IgM is the first type of class of antibody that a newly maturing B cell will make. As the B cell goes through and starts doing what's called hypermutation, once it comes into contact with an antigen that its antibodies can bind to, hence it can bind the B cell receptor, it starts going through mutations to make that variable region just a little bit better at binding an antigen. But it also triggers what's called class switching. So instead of IgM, the initial, depending upon where the B cell is located and where the antigen's coming from, IgM could become IgG, which is the primary antibody you find floating throughout your body, your bloodstream. Or it can become IgA, IgE, or IgD, depending upon what needs to be done. Is it going to be in the blood? Is it going to be in the tissues? It has to be something that has to be secreted across the membrane or a mucous membrane, like in your tears or your saliva. Different classes have different roles because of their different shapes, which is based on the why we talked about in the last couple of slides. It's just, is it, as you look like with IgE, does it have more immunoglobulin domains to stretch out the FC portion? Is it the pentamer thing like IgM? IgA, which has a J chain joining two immunoglobulins together, two antibodies. So it all depends on where those antibodies need to be will determine which class is going to come. Is it always foolproof? No. Different B cells are going to produce, different classes, even though they're all responding to the same antigen. And, a B cell may class switch and inadvertently go to IgE. Well, turns out that the antigen is actually in the blood, and IgE is for secretion in the tears across the mucous membrane. That B cell that's class switched to IgE, well, it's rarely, if ever, going to get triggered or, reactivated later. And as I alluded to just a minute ago, antibodies have multiple functions. Opsonization, it means if you have something, a cell, a bacterial cell, a virus, parasite or a parasitic larvae in the tissues, the antibodies will start to coat whatever it is that's not you. So they start to form this coating of proteins so that if it is a virus, it can't come into contact with one of your cells, can't force uptake. If it's a bacteria, can't come into contact, can't force its own uptake to hide. Neutralization, if it's, a foreign protein, such as a toxin or a toxoid. Complement activation, as we talked about in, previous lectures on the innate. Complement, specifically the classical pathway, needs antibodies bound to the surface of something. Antibodies will start complement activation, triggering C1 cleavage, which does 2, 3, and so on. And then what's referred to as ADCC. You get enough antibodies coating and binding to something, it's going to start changing, it's going to start altering plasma membranes, it's going to start, destruction from the plasma membrane in on something not you.